Of the molecule where there are three particular bases that do not vary. I think there's importance to the general three-dimensional shape of these parts. I think the amino acyl tRNA synthetase is grabbing by a bunch of these different knobs to figure out that it has the right tRNA. But the other important thing is the other end. At the 3' end, we have the synthetase has already correctly identified and attached the right amino acid, so that there is a correspondence between the amino acid and the anticodon. And so that is sometimes they call this the adapter molecule. If you're a mechanic and you know about adapters, you've got the nucleic acid language at this end, and that has been converted into an amino acid language at the other end of this adapter. Francis Crick suggested there must be an adapter, and then he went out and found one. So he's the much smarter part of that team. I like this diagram. It gives some attention to the synthetases that are responsible for putting the correct amino acid and the correct tRNA species together. Sometimes we forget them. Ribosomes we think about a lot. There's lots of protein portions. There's lots of ribosomal RNA portions. Together they make up subunits that come together to form the completed ribosome. Maybe the most interesting thing about this, when I was a student, they told us that the catalytic portion of the ribosome was some protein in the large ribosomal subunit, and it catalyzed the formation of the peptide bond. That turns out to be wrong. It is one of the rRNAs that is in the large ribosomal subunit, which is actually the catalytic portion of this multi-part enzyme. So then they had to start saying enzymes can't be proteins or RNAs. So this is one of the most ancient and important catalytic functions, and we didn't even know that an RNA was responsible for that. So it broadened our minds a little bit. You guys maybe have heard that. Enzymes can be proteins or RNAs. And an example of an RNA enzyme might be the large ribosomal subunit itself. The code like this you'll have in your hand. You may have to remember that AUG is when translation will start. AUG can be in the middle of a gene, but it must be at the beginning of the coding region. We said differences between initiation in prokaryotes and eukaryotes is the only difference that we have to keep track of. What we're binding to in the prokaryotic world is the Shine-Dalgarno consensus sequence. That sequence in the RNA will form hydrogen bonds with a complementary anti-parallel stretch of that, I think it's the 18S ribosomal RNA that's in the small ribosomal subunit. So that's what we're binding to. We're binding to the Shine-Dalgarno, which we said is sometimes called an example of an internal ribosomal entry site. And so I do continue to hear that term iris in upper level courses and in real papers where we're talking about genetic engineering, for example. Sometimes you engineer your genes to have an iris because it's awfully convenient, but it's not something that eukaryotes do naturally. That initiator tRNA is the only one that comes to the coding region and is already in the P site. Sometimes that's a trivial piece of information. The initiator tRNA is the only one that binds at the P site. Remember, all the other tRNAs are jumping into the A site. Then there's the peptide bond, and then everything moves down a click. So the initiator tRNA is special. It always has formylated methionine in a prokaryote, and it has anti-parallel complementary bases for its anticodon. And so between the Shine-Dalgarno and the ribosomal RNA and the codon-anticodon combination, that's what's putting this in the correct frame to actually start translation. Once we're in the correct frame, large ribosomal subunit comes. A site, I guess, would be over here, and as soon as we get another amino acid sitting next to this one, that catalysis function must be located right about there. In eukaryotes, this artist does everything upside down, but the 5' on the left, 3' on the right, we have the small ribosomal subunit already attached at the P site to the initiator tRNA, which is bearing MET, not formulated MET. We don't bind to anything called Schindel-Garno. There is no internal ribosomal entry site. Ribosomes enter at the 5' methyl G cap, and then they cruise down until the initiator anticodon hydrogen bonds with the start codon. And so the start codon, the appropriate frame, is discovered by a hydrogen bonding interaction. When we're in the right place, then the large ribosomal subunit will bind. And there's our A site ready for all the other tRNAs to enter. So the initiator tRNA is the only one that comes to the party in the A site. This idea of an internal ribosomal entry site is strictly prokaryotic. And here is a diagram of a typical prokaryotic mRNA. They're showing 5' end with the phosphates out there, as you'd expect, a 3' end with nothing decorating it. It was just produced when the RNA polymerase quit after it got to that terminating hairpin, followed by a string of U's, and then just fell off. But what we find in a prokaryotic mRNA is a Schindel-Garno right there, a start codon right there, and then the ribosome can translate until it hits a stop codon and then quits, producing that protein. The very same mRNA must have a Schindel-Garno right there, start codon right there. The ribosome can cruise down until it's told to stop, and it will produce that protein. There's a Schindel-Garno right there, an AUG right there. Ribosome will jump on, cruise down, translate until it's told to stop, and we'll get that protein. And so this is a term, I got him upside down. I just made this 5 minutes ago. That means it's posted wrong. I've got to go home and get the posted there. Oh, now I can't see it at all. I'll put it down below, just for now. So these coding regions that lead to different proteins, are those 3 different genes? Maybe. It was a semantics argument. But this mRNA was transcribed downstream of a single promoter, a single -10, -35. So maybe that's one gene, one promoter, one gene, except there's 3 gene products. So they kind of avoided this by calling this whole setup an operon. And prokaryotic genes tend to be organized into operons. An operon is a genetic unit with a single promoter leading to a single mRNA. However, that prokaryotic mRNA is said to be polycistronic. Cistron was the original word for gene, and there are multiple gene segments, multiple coding segments, multiple gene products that can be resulting from this single mRNA. So gene structure in a prokaryote, genes are organized into operons so that that one promoter leads to the expression of the entire transcript that leads to the co-expression of all 3 of those proteins at once. And what we find is these 3 proteins, alpha, beta, gamma, are likely to be required to carry out the exact same biochemical pathway. You have a substrate, you want to get a product, you may need 3 enzymes to get there. They all get expressed on the very same polycistronic mRNA. That mRNA is encoded by a single operon, a single promoter. And it allows prokaryotes to be highly organized, and if you need 3 proteins at once, express them all at the same time, rather than figure out how you're going to coordinate the expression of 3 different things. Eukaryotic mRNAs can't be that way. They are monocistronic. There can only ever be one coding sequence. And it has to do with this idea of we don't have irises. Since eukaryotic ribosomes bind to the one and only 5' methyl G cap, they're going to cruise down, translate till they get to a stop codon, and then fall off. And I don't care what you stick downstream of that, it's never gonna get translated. You can put a bunch of things down there and they're all gonna be ignored because the ribosomes can only enter at the tip, they cruise down, begin translating, and then finish. And then that's it. One gene product, one promoter, one transcript, and monocystronic is the word. And sometimes students are asked, why are eukaryotic mRNAs monocystronic, whereas prokaryotic mRNAs are polycystronic? And it has to do with ribosomal entry. Ribosomal entry is only at the 5' methyl G cap, so only one product can be translated, whereas because of internal ribosomal entry sites, the Shine-Delgarno sequence, multiple internal ribosomal entry sites can allow translation of multiple protein products. So polycystronic, monocystronic is an important piece of terminology. We worked with this transcript last time. At this point, we're thinking about elongation, and we weren't concerned whether this was prokaryotic or eukaryotic. We imagine binding happens somewhere up here, and that the start codon, this must be the very first AUG after ribosome binding. So methionine there, serine there, ASN there, alanine there, cysteine there, and so on and so on and so on and so on. And we expect the protein is going to be several hundred amino acids long, some over 1,000. We should mark this as the N terminus, and the other end is the C terminus, having to do with the monomer structure that we already know, with an amino group, an N group, and a carboxy, a C group on the other side of the amino acid monomer, and therefore on the sides of the polypeptide chain as well. Then we talked about changing a base, like this one. So I'm changing that last base. That has no effect on the amino acid specified, and we saw that on our genetic code. And if you look around the genetic code, beyond this one example, mutational changes in that third position in a codon frequently make no difference. There's a whole bunch of boxes on the genetic code where it doesn't matter what's in that third base, all of them spell arginine. It doesn't matter what's in that third base, they all spell proline. And so that is something that we tend to expect. If I change the second base in a codon, however, that almost always is going to cause an absolute change in amino acids. So that's the missense mutation. A change in the DNA leads to a change in the RNA, leads to a single amino acid substitution in the protein, which may or may not be important. Put that back if we change something else. Certain changes that we make can lead to the creation of a stop codon that will truncate the protein and is likely to have a devastating effect on the protein's ability to function. We might think of nonsense mutations as almost always loss of function mutations. Missense mutations, maybe they don't cause much of a functional difference, or maybe they cause a huge functional difference. It matters if it's an important change or if it's an important amino acid, as we discussed last time. Students are almost always asked to reveal that they can use appropriate vocabulary to describe that they know the difference between a missense and a nonsense mutation. I'm sure it'll be on your test. Finally, if we subtract, as I think I'm doing here, I'm just taking away that U. Presumably because a T has been lost in the coding strand of the DNA gene. That's going to change the reading frame from that point on. Since we read in sets of 3, we've adjusted the frame by the loss of that single base. It'll be the same effect if we added, like in my mouse, an extra base is going to upset the reading frame and cause wrong amino acids from that point on. If we added or subtracted 2, or 4, or 5, or 7, any non-multiple of 3, a frame shift mutation is what we expect to ensue. So a whole bunch of incorrect amino acids, and probably an out-of-frame stop codon will be encountered before too terribly long. I think on average you might go 20 before you accidentally bump into you are away, you go away, you are gone, the stop codons. So silent missense, nonsense frame shift. Those are the important mutations that students are always taught as soon as we hear about coding regions and translation. Keep in mind, there are many other parts of a gene than just the coding regions. So I want you to not just consider these are the only kind of mutations that there can be. Those are point mutations in coding regions. But we talked about the promoter as having a particular consensus sequence. What if you mutated that? I don't think it would be described by any of those words. A promoter defect means there's no what. So now you reveal to me that you know what a promoter is for. It's for the binding of the RNA polymerase. It's important for the initiation of transcription. So with a promoter defect, there's going to be no RNA produced. Without any RNA, there can't be anything translated either. So a promoter defect, we kind of get no gene expression either at the RNA level or the protein level. And I think that's what you want to think about. We will ask questions about these kinds of defects, and some of them will get rid of both the transcript and the protein. Some of them will screw up the transcript but not the protein. Some will screw up the protein but not the transcript. They don't always travel hand in hand. And so what this question is really asking is, do you know the difference between transcription and translation? And everyone says, yeah, and then they get this question wrong. Do you know the difference between RNA and protein? And everyone says, yeah, except then they get the question wrong. So the question is, do you know the difference between these processes? So you want to go through this and not just say an easy yes, but can you really think it through? Splice donor, splice acceptor defects. Well, to answer the question, you have to reveal that you know what those are for. This might be how we describe a eukaryotic gene divided into those exons. There ought to be a splice donor here, a splice acceptor there, a splice donor here, a splice acceptor there. And maybe the spliceosome is supposed to put exon 1, exon 2, and exon 3 together. That's what's supposed to happen. But now we're asking you, consider a mutation in that sequence that maybe defines a splice donor. That's gone because I've changed the base in that consensus sequence. Now what's going to happen? Let's think about that in red. Now, when this primary transcript is produced, the spliceosome comes along, is looking for a splice donor, does not find one. Therefore, it takes exon 1, leaves intron 1 to 2. That's now connected to, or was always connected to exon 2. And then we finally find a splice donor and attach it to a splice acceptor. Well, that's not right. This is exon 1. We're keeping the intron. We're retaining an intron due to this splice donor defect. Then we have exon 2 connected to exon 3. Well, what's that going to do at the RNA level? We've just described it. We will be retaining an intron that we should have been throwing away. So that answers that question. What's the effect on the transcript? What's the effect on the protein? Well, now we have to think about translation. Maybe our start codon's there. Maybe there's a bunch more codons. Now we're going to be translating right through this intron. That wasn't supposed to happen. So we'll have the correct amino acid sequence. Then we'll have crazy stuff. Remember, these introns tend to be thousands and thousands of bases long. Are we going to make it all the way across the intron before we find an outer frame stop codon? Not too likely. Let's imagine maybe we do. Maybe this is a small intron. If we do get normal stuff, weird stuff, when we start translating here, what's the chance it's in the right frame? I think 1 in 3, because there's 3 reading frames. 2 of them are wrong. So maybe I'm in the right reading frame, maybe I'm not. Depends upon exactly how many bases are in here. So maybe I keep getting weird stuff if I've even gotten that far. So there's a bunch of possibilities for what's wrong with that particular protein. Let's change the question. That was splice donor. What if we mutate instead splice acceptor? It's not the same answer. We can stay with a simple diagram. Maybe we're supposed to have exon 1, exon 2, exon 3 to keep it simple. Splice donor, splice acceptor, splice donor, splice acceptor. What if we mutate a splice acceptor? Now the spliceosome comes along, looks at this primary transcript, finds a splice donor, attaches it to the next splice acceptor that it finds. And that one's gone. It doesn't find one till maybe way over there. We have certainly changed the transcript. We can be descriptive. We have skipped an exon inappropriately, in all likelihood. And again, what's the effect on the protein? Well, if we start translating here, things will be fine, and then we skip a whole big section of the protein, and then there's only a 1 in 3 chance when we get over here that we're in the right frame. So we might start to get weird stuff. So at the very least, we are missing an entire domain of the protein. So domain is the word for a section of protein that often has a specific function. Or we're going to have normal stuff and then weird stuff, maybe even with an early stop, if we're out of frame, which is a 2/3 chance of that, I think. Yeah.
If you have two splice donor sites, which one is going to bind to the splice acceptor?
I think the way it goes, so that SNRP, the U1 SNRP, this is all happening in the nucleus, so the RNA polymerase is kind of cranking out this transcript. First thing that pops out is the splice donor, and so I think the U1 SNRP is going to grab that. So you'll grab your first one.